Hello everyone, my name is Lucia Castelli and I want to present my final project for CS50R. It's called degfind. So degfind is the name of the project and the package. My GitHub username is Adorcas, my edX username is Lucia_Castelli21. I'm from Buenos Aires, Argentina, and today's August the 13th, 2024. I will now briefly explain what differentially expressed genes, or DEGs, are. They're a tool that I need for my master's thesis, so that's what drove me to take the R course and to build this specific package. DEGs are genes that show different levels of activity when comparing two groups, for example, a healthy person with someone who is ill. By analyzing biopsies, scientists can identify which genes are more or less active significantly in the ill person compared to the healthy one. Thus, we can infer which biological pathways are upregulated or downregulated in each group. These differences can help us understand the illness behind the scenes and find potential treatment targets. I owe my understanding of the pipeline for this analysis basically to two sources, one of them being Daniel Biding's DIY Transcriptomics online course, and the other being the guidance of my thesis director, Joaquin Merlo, here at the IBIME Institute in Buenos Aires. For this final project, I have built 8 functions. The idea is to later on add more, but these 8 are the ones I'm going to show you right now. After installing and loading all the necessary things, we have our sample data, which must look like this. All the rows are the gene names and the columns are the sample names of the normalized data. And the study design table must have a sample ID column and a phenotype column. With this, we can do all our analysis. First, we can extract the sample names. So here they are in a vector. We can do the same with the gene names. Here we have the gene vectors. We can also plot the data quality and generate a violin plot, which we can save with our custom name. We can also print it here, or choose not to if we change this parameter to false. Then we can make a linear model. The first element of the linear model will be the linear model itself, and the second one will be the Bayesian statistics. So this is very useful to make a top table, which is similar to that in Lemma, but not quite. We have less columns and a new column here. The rank one is to account for the high expression genes and the low expression genes. We can also extract the differentially expressed genes and generate a Venn diagram here of the ones that are upregulated and downregulated. So here we have the names of the DEGs. We can also make a volcano plot, which we can save with a default name, or we can change it as well. But this is just an example. So here we have the log fold change and the minus log 10 of the adjusted p-value. So these are the downregulated genes and these are the upregulated genes. We can choose a p-value of our choice or a log fold change of our choice, and it will color the dots that are significant. Then the last function is degtable. It saves differentially expressed genes table in our files. Here we have it, degtable.csv. And we have the gene ID, log fold change, p-value, adjusted p-value, and rank, and this is for the reports. My name is Lucia, and this was CS50R.